Good morning, everyone.
Good morning.
Good morning.
Welcome back. Okay, welcome back, everyone. Just taking a moment to allow a few additional folks into the class here. Okay, so we're going to continue our conversation this morning with Chapter 3, specifically focusing on processes that cells rely on to move cargo through the plasma membrane. That's our primary talking point this morning. But before I continue, are there any questions you have at this time? Course content, schedule, anything you would like to ask?
So our first exam is Monday, correct?
No, let me get the syllabus.
Okay. And our exams for regular anatomy and the lab are separate. Yes?
Yes, okay, so your first exam of the semester is next Wednesday. That will be a lab exam. And your first lecture exam of the semester is a Monday, October 11th, okay? So
Okay.
I want to encourage you to print out your syllabus if you don't have it printed out, and keep a record for yourself.
Thank you.
Okay, other questions? Okay, gang, as I said, good morning to you and welcome back as we rub the sleep out of our eyes and try to think about some technical things. Okay, let's get started here this morning. I'm going to talk to you about a very important topic in Chapter 3. Okay, and I want to remind you about a resource that you do have access to through Wiley Plus. In today's class, I will be showing you two alternative videos on transport processes that move cargo through the membrane. One is called membrane transport passive processes. The other is called membrane transport active processes. Don't forget that you have access to these resources in Wiley Plus. Let me show you that real quickly at this time. So if you're in the Wiley Plus website, in the lecture portion of Wiley Plus, right? If you go up to Wiley Course Resources and click on that in the upper left, it will open up, excuse me a second while I wait for my window it will open up this tab where you have course-wide resources. If you open up course-wide resources and click on 3D physiology, you'll end up with this page right here. These are the two videos that I'm highlighting in your notes that I want you to use again during your private study time. Okay, I want to show them to you today, but it will be helpful to you to see them again when you're studying. Okay, so just don't forget about all these things that are present. There's a lot of balls in the air, a lot of things swimming around in your head right now. Don't forget, there's other resources on Wiley Plus that can help you. Okay, let's begin. All right, so last class, we spent all of the class talking about the anatomy of a typical cell. Well, we described many of the parts, not literally every part, but many of the parts of a cell. Well, another major talking point of Chapter 3 is discussing, well, how does the cell get material into the cytoplasm? How does the cell push material out of the cytoplasm? And in both cases, whether you're entering the cell or exit the cell, you have got to get through the plasma membrane, right? Because the plasma membrane defines the border of the cell. So our talking point today is moving cargo through the plasma membrane, and we're going to start talking about this very important concept. Let's start with the topic of membrane permeability. You know, that word permeability suggests that you can get through the membrane, but there will be times when the membrane is actually impermeable and you cannot get through the membrane, okay? So we're going to make some general statements here. Okay. First statement. The phospholipid bilayer of the plasma membrane is permeable. It will allow materials to get through it, which have the following characteristics. They are nonpolar, they are uncharged, nonpolar and uncharged, okay? The phospholipid bilayer of the plasma membrane is permeable to nonpolar, uncharged molecules, okay? Let me highlight that. Nonpolar, uncharged molecules, generally speaking, can get through the phospholipid bilayer. Let me give you a couple examples of these molecules. Examples of molecules that are nonpolar and uncharged would be oxygen, would be oxygen, carbon dioxide, and steroids. Okay, oxygen, carbon dioxide, and steroids. Okay, quick question coming into the chat window for you. Okay. What is the molecular formula for oxygen that we breathe into our bodies from the air? What is the molecular formula? Do you know what the molecular formula is? Okay, it's awfully quiet out there. I think I can hear some crickets chirping. Okay, all right. So Noah's gotten us an answer that we can use. Thank you, Scarlett, and thanks everybody for trying to answer this. Okay, what I want to point out is that the kind of oxygen that we're taking into our lungs is O2. Okay, O2. All right, having a hard time writing today. There we go. So let me make a talking point here, okay? Be careful when talking about oxygen because there are many forms of oxygen. For example, okay, you can have elemental oxygen. And when I say elemental oxygen, I'm talking about an individual oxygen atom as it is defined on the periodic table of elements. Okay, so if you hear somebody talk about oxygen, unless it's quite plainly obvious what version of oxygen he or she is talking about, you might ask, well, what form of oxygen are you referring to? Because elemental oxygen is the individual oxygen atom of the periodic table. We also have atmospheric oxygen. Okay, atmospheric oxygen is the oxygen we've just written down. It is O2. This is the form of oxygen we're breathing into our lungs. Okay, this is 2 atoms of oxygen, okay, O2, covalently linked. Let me say it this way, linked by a covalent bond, okay? There are even other forms of oxygen that we could talk about. Let me ask you this. What is the molecular formula here in the chat window? What is the molecular formula for ozone? What is the molecular formula for ozone? Okay, thank you. No, it is in fact O3. Ozone is an interesting material. It's actually highly toxic to life, right? If you were to bubble O3 gas into a water supply, it would basically kill everything present. Okay, so there are many different forms of oxygen, and we need to be aware of the specific version we're talking about at any moment of time. Now, for our course, Anatomy and Physiology, typically when we're talking about oxygen, it's typically in the context of air that you're inhaling, and so you can think of it as being atmospheric oxygen. Okay, so first statement, the phospholipid bilayer of the plasma membrane is permeable to nonpolar uncharged molecules. We're going to dissect that statement a little bit from now, okay? Next statement, the phospholipid bilayer of the plasma membrane is impermeable. It will not allow the passage of, it is impermeable to ions, all right? Sodium ions, chloride ions, potassium ions, hydrogen ions, as well as and uncharged polar molecules, okay? Uncharged polar molecules. So let me give you some examples here. Once again, sodium ions, chloride ions, potassium ions, hydrogen ions, none of these things are going to get through the phospholipid bilayer on their own. They're not going to be able to get through. They're not going to be able to squeak through the phospholipids of the plasma membrane. They can't, because a lot of times the electric charge actually repels from the surface of the phospholipid bilayer. In addition, there are other molecules that are not charged, but they're still polar, and they also have a hard time getting through the plasma membrane. So an example of that would be something like glucose, right? Glucose is a monosaccharide, as you know. It is a simple sugar. It's a type of carbohydrate, okay? So these sorts of things are impermeable with regard to the plasma membrane. Now, like anything, there's often some exceptions, okay? So let me say this, okay? One notable exception to this rule is water. You know, water is uncharged, but it's polar. Water is uncharged and polar. But it still can get through the plasma membrane. Okay, so the fact that we have to describe things this way in your textbook represents two realities. Number one, this is what when I say we, I mean this is what science observes. These things are true because we can test them, we can measure them, we know that this is true. But we can't always get a very big unifying theory to understand why certain things move through and other things don't. And we always have to put a little fine print, right? The fine print at the end of the commercials that says, here's a rule that works, except in these scenarios, right? So there's some things in our scientific understanding of what enables material to move through the plasma membrane is still a little bit lacking, if we're completely honest, okay? All right. So one notable exception to letter B is water. And I will mention to you also, if we highlight the language of letter B, the phospholipid bilayer of the plasma membrane is impermeable to ions and uncharged polar molecules, okay? For substances like glucose that cannot get through the membrane, or substances like chloride ions that cannot get through the membrane, well, how does the cell acquire these objects? Because they have to get into the cell at various times, and the answer is through the assistance of transmembrane proteins. You may recall we talked about the anatomy of the plasma membrane. Again, figure 3.2, roughly on page 65 of your book. To get these types of materials through the plasma membrane, specialized transmembrane proteins are used, okay? So these materials, generally speaking, for letter B, ions, glucose, similar things, cannot float in between the plasma membrane phospholipids. That can't happen. It doesn't work. So how does the cell acquire these materials? These materials move through transmembrane proteins. Okay. All right, let me make one more statement and then we're going to come back and talk about the language here. Letter C. Very, very large cargo. So let me give an example of what would be considered large cargo. Large cargo would be something like a protein. Most proteins are giant compared to a small molecule like diatomic oxygen, right? Diatomic oxygen is about as small a molecule as you can get. 2 atoms of oxygen, that's about as tiny as a molecule will get, okay? So it has a good chance of getting through the membrane because it's so small. But if you have large molecules with hundreds to even thousands of atoms, they're much too large to get through the plasma membrane simply by floating through the space in between the phospholipids of the bilayer. So how does the cell acquire them? Okay, huge molecules like proteins cannot pass through the plasma membrane, but can still enter a cell through the process of endocytosis. Okay, so we're going to be talking about endocytosis later on. It can enter the cell through the process of endocytosis and exit a cell through the process of exocytosis. Okay, so just be aware of these things during your private study time, that these processes, these topics, these terms represent valid testing points on your exam. Okay, your first lecture exam is now about 2.5 weeks away. All right. Do you have any questions right now? Okay, I suspect you should have some questions because I suspect you do not understand this completely. So sometimes you don't even appreciate what you don't understand. And so let me see if I can tickle you a little bit here. Okay. So we have this language up here, nonpolar, uncharged. We have this language, ions and uncharged polar molecules. Okay, there's a lot of language in there that really means specific things and can be confusing. Okay, so let's talk about this. Clarification of terminology. Okay, let me put this into the chat window. Here we go. What is the difference? What's the difference between polar, nonpolar, and charged, uncharged? Because it seems like that could kind of be the same thing, but it's not. What is the difference between polar / nonpolar and charged / uncharged? I have found over the years that students do confuse these ideas. Anybody want to take a shot at this? Okay. Okay, Noah has an interesting statement in the chat window. Polar means electrons are skewed one way. I very much like that, Noah. I do like that. So Noah or anybody else, how would that be different from, say, the term charged? Jordan, you say nonpolar is shared equally. That's true too. I love that statement. I love that statement. Okay. All right. So let's do this. Comparing polar, nonpolar, and charged, uncharged. Okay. All right, so let's take charged and uncharged first. With charged and uncharged, let me give you something to think about, to remember about. Remember when we talked in chapter 2 about sodium chloride, table salt, okay? Right? The whole foundation for the attraction between these two atoms is that one atom, in this case sodium, donated an electron to chlorine, and chlorine accepted the electron. So when we're talking about charged and uncharged, we're talking about an imbalance in the number of protons and electrons present in the nucleus of each atom, okay? Or if I could say it differently, you know, when we're talking charged and uncharged, this is a phenomenon created when one atom gives away electrons and another atom accepts electrons, okay? Okay. Each individual atom in the molecule, in this case, each individual sodium, each individual chlorine, each individual atom within the molecule will then have an imbalance in their number of protons and electrons. Okay, so before we go any further, question in the chat window. Are my comments for charged/uncharged making sense? Is there anything else is there anything you'd like to ask about that? So the idea when you have a charged atom, you have a charged atom because literally there's an imbalance between the numbers of protons and electrons. Why is there an imbalance? Because that atom just gave away an electron or that atom just accepted an electron. Okay. Now, polar and nonpolar, it seems like we're talking about charge too, but it's a little bit more subtle. Okay. Polar and nonpolar. For this one, consider the OH bond that exists within water. Okay, because this is how we talked about it in chapter 2. In this scenario, there is no imbalance in the number of protons and electrons for each individual atom. There is no imbalance. But where the partial charge comes into play is that one atom is pulling electrons towards itself a majority of the time, and the other atom is losing its electrons the majority of the time. But this is still a sharing. It's just an unequal sharing, an unequal or equal sharing, polar versus nonpolar. Okay, this refers to a scenario where partial charge, okay? If you say something like sodium chloride is a charged substance, it's because there's a complete charge because the atom has donated or accepted an additional electron. That's a full change in charge because of the imbalance in protons and electrons. With polar and nonpolar, there is no imbalance in protons and electrons. What there is a slight imbalance in how the electrons are being shared, okay? Partial charges are created by the unequal or this refers to a scenario where partial charges are created by the unequal sharing of electrons in the case of polar, okay? So, you know, if you were to ask which scenario represents a more powerful charge, which scenario represents a more powerful charge, maybe we should ask that in the chat window. Folks, you answer that. Which scenario represents a more powerful charge? Is it the charged and uncharged terminology or the polar and nonpolar terminology? Okay, so Rochelle says charged and uncharged, so does Noah. How about the rest of you? Do you agree? Do you disagree? Okay, I hope you agree, and I can hope you can understand the subtle, and it is subtle, but it's still important. It's a subtle difference here, okay? Let me just make one final comment. Each individual atom will not have an imbalance in the number of protons and electrons. Okay, so, you know, it's worth your consideration when you read these statements up here to consider what it means to be nonpolar and uncharged, or uncharged and polar. Okay, what does that really mean? Okay. All right. So one last thing here under membrane permeability. We need to talk about gradients. Okay, gradients. Let me make this letter D. Oops. Okay, wrong place. Okay. All right, gradients. Okay, gradients as a talking point is on page 68 of your textbook. There's a heading for a column of text that says gradients across the plasma membrane. Okay. The ability of material to move in one direction or another relies on the formation and maintenance, quite frankly, of gradients. You know, a gradient is an area where you have a bunch of something and another second different location where you have less of something. Okay, think about a seesaw. Okay, right now, my right arm happens to be up, so that's the high area of the gradient. My left arm is down, that's the low area of the gradient. Okay, so that distance between my right hand and my left hand is a gradient. Okay, so basically there are three types of gradients that the cells rely on to move cargo. First is a concentration gradient. By concentration gradient, we're talking about a difference in the concentration of a chemical, the difference in the concentration of a molecule between two locations. Well, those two locations are going to be inside the cell versus outside the cell. Difference in concentration between two locations inside and outside cell. Okay, so if you had 50 molecules of a specific amino acid outside the cell, and you had 25 molecules of those amino acids inside the cell, then there would be a tendency for those molecules to enter the cell, because you go from high outside the cell, 50, to low inside the cell, 25. Okay, so that gradient, wherever you have an excess, the excess is going to kind of move towards the area where there's fewer to try to balance out the gradient. Okay, not only do we have concentration gradients, we have electrical gradients. Okay, for an electrical gradient, we're talking about a difference in electric charge between two locations, inside and outside the cell. Difference in electrical charge between two locations, inside and outside the cell. Okay. And then the third type of gradient, which frankly I believe is probably the most common type of gradient that cells actually utilize and maintain to move their cargo, is what's referred to as an electrochemical gradient. And by electrochemical, that refers to the combined forces of the concentration gradient and electrical gradient working together as a team, working together synergistically. Okay, so the electrochemical gradient, this refers to the combined influence of chemical, meaning concentration, and electrical gradients on the movement of particular ions. Most notably would be examples like hydrogen ions, or potassium ions, or calcium ions, that kind of thing. Okay, so right now we're talking in generalities, but you're going to find throughout the entire semester there's going to be numerous times, particularly when we get to the nervous system at the end of our semester together, the bottom third of our time together, we're going to be talking about electrochemical gradients and how they're involved in the maintenance of what's called a membrane potential with neurons. Okay, so we're just introducing it to you today as something to start thinking about. And we'll actually start using it more and more as we get into other topics in the course. All right. Gang, do you have any questions you'd like to ask at the moment? Okay, I know this can be intense. I mean, it probably is intense, but you can do it if you wrestle with it a little bit each day. Slowly the fog dissipates, if you will. Okay, so what I'm going to do next is start talking to you about passive processes of moving cargo through the plasma membrane. And so I'm going to make this number 2. Make this number 2 in our notes here. And I'll just maybe make a heading here because passive transport processes. I'm going to show you a video. I'll show you a video on this from Wiley Plus, the same one I just showed you that it exists at the start of the class today. Show it to you, then we'll talk about it in some detail, okay? Let's do this. Let me get this queued up so we can watch it together. Okay. All right. Here we go, gang. Watch, enjoy it, and we'll have something to talk about here. Here we go.
The 60 to 100 trillion cells of the human body are surrounded by extracellular fluid containing substances needed by the cell, such as oxygen, ions, nutrient sources of energy, and ingredients for cellular functions. This fluid is also the location for disposal of wastes and carbon dioxide. All of these substances need to be transported across the plasma membrane. Active processes utilize energy to move a substance across the membrane, generally from areas of lower concentration to areas of higher concentration. Passive processes require no energy to accomplish this movement. Gases, ions, and nutrients are transported this way. The structure of the plasma membrane plays a large role in determining the specific transport process utilized by the substance. The plasma membrane is constructed of 2 layers of phospholipid molecules. Each phospholipid has a polar head and two nonpolar phospholipid tails. The polar heads face the extracellular fluid or the cytosol. Their nonpolar lipid tails face each other. This arrangement prohibits electrically charged, or polar, molecules from passing through the lipid bilayer of the plasma membrane. Small uncharged and nonpolar substances can pass through freely. The kinetic energy of its atoms or molecules causes a substance to move from an area of high concentration to an area of lower concentration. The difference between the two is referred to as a concentration gradient. If the substance is charged, the distribution of positive and negative charges on opposite sides of the plasma membrane also results in an electrical gradient. The combined influence of differences in electrical charges and in chemical concentration is referred to as an electrochemical gradient. Passive processes occur when a substance travels down its electrochemical gradient, either unassisted or with help of a transport protein. Simple diffusion is a passive process involving unassisted movement across the plasma membrane. Nonpolar substances and small uncharged substances, such as oxygen and carbon dioxide, pass through the phospholipid bilayer with little resistance. Ions and polar molecules cannot move through the phospholipid bilayer via simple diffusion. The passive process of facilitated diffusion uses protein transporters to facilitate the movement of substances through the plasma membrane. Channel-mediated facilitated diffusion involves an ion channel, and carrier-mediated facilitated diffusion uses a carrier protein. Channel-mediated facilitated diffusion requires the presence of a tube-shaped ion channel across the plasma membrane. Each channel is selective, allowing only certain ions to pass through. Common ion channels allow passage of sodium, chloride, potassium, or calcium ions. Most ion channels change shape, acting as if they have gates that open and close to allow or restrict ion passage. In another type of ion channel, a leak channel, ions pass slowly but almost continuously through the channel. During this facilitated process, substances diffuse down their electrochemical gradients. In carrier-mediated facilitated diffusion, a carrier protein transports a solute across the plasma membrane. Each carrier protein possesses internal binding sites specific to a particular solute, such as glucose. When those binding sites are occupied, the carrier protein changes shape, opening to the opposite side of the membrane and allowing the solute to pass. A final passive process is osmosis, the movement of solvent rather than solute. In osmosis, a solvent, such as water, moves down its concentration gradient. A higher concentration of solutes on one side of a membrane means a lower relative concentration of the solvent on that side. As the quantity of solutes increases, the osmotic pressure also increases. In general, as solute concentration is increased, so does the tendency for water to move towards those solutes. Osmotic pressure is a measure of the force exerted as water is drawn across the plasma membrane in an attempt to equalize the concentration of solutes.
Okay, folks. So, hopefully you found that somewhat helpful to begin thinking about this topic. Okay, we need to talk and put into your notes many of the things that the animation was describing to you. Okay, so don't forget, you have access to the same video through your Wiley Plus course resources. Do you have any questions you'd like to ask simply from having watched that video one time? Okay, everybody's so quiet. All right, passive processes. When we talk about passive processes, let's be sure we understand what passive means. Passive means that some kind of cargo is moving across the membrane, but the cell is not burning any of its own biochemical energy to move that cargo. Okay, it's kind of like a free ride on the bus. You're not spending any of your money to go on the bus because somebody else is paying for it. Okay, so passive means in this context, passive implies that the cargo is moving from kinetic energy, which is not supplied by burning fuel inside the cell. Okay, in other words, the cell is not consuming ATP in order to move the cargo. Now, some of you might remember from other sources of education you've had that kinetic energy is the energy of motion. And while I like that your video talked about kinetic energy, that kind of avoids the deeper question of, well, how do these molecules get to move at all in the first place? So let's talk about that. Or I don't want to spend a lot of time on it, but here's a question in the chat window. There it is. What is the source of energy that allows molecules to move without the cell burning fuel? Okay, Karen, diffusion is not the source of energy. Diffusion is the movement of the molecules, but the molecules don't move unless there's an energy source. So where's the energy? Okay, yeah, okay. The energy is kinetic, but it's not ATP, Rachel and Shay. It's not ATP here. The cell is not burning ATP in order to move this cargo. Okay, that's why there's a bit of confusion here. This is a passive process, right? This is a passive process where molecules are pushed into motion. Well, what pushes them into motion? When they're in motion, that's the kinetic energy. But what pushes them into the motion? It's not ATP. It is not ATP. Folks, it is heat energy from the environment. Heat energy from the environment is what moves the molecules, okay? Think about what happens when you crack an egg, crack the shell of an egg and put the content of the egg into a frying pan, turn on the burner of the stove, and there's heat energy, right? Doesn't the egg cook? Well, all those molecules in the liquidy white egg are going to move around, okay? So it's heat energy from the environment, which ultimately moves the molecules, which imparts kinetic energy to them. And that's what allows them to move without the cell burning any ATP, okay? So kinetic energy is energy of motion. This is supplied by the surrounding heat energy of the environment. Okay. Now, when we're talking about passive processes, we're going to be talking about moving some form of cargo from an area of high concentration to an area of low concentration. That's the common thing here. Two common things for all passive transport processes. The cell does not burn any ATP, and we're moving some kind of cargo from a high concentration area to a low concentration area. Typically through the plasma membrane, okay? So there are multiple types of passive transport processes that are used by the cell. We'll make a list here. First, we have simple diffusion. Okay, you can look at your book as well as I can. Figure 3.4 on page 69 is a good illustration of simple diffusion. If you were to take, well, you probably have all, at some moment in your life, even if just a childhood memory or something, you probably have all had some experience with coloring Easter eggs. Coloring Easter eggs, okay? What I'm saying is, if you want to color an Easter egg, you take a hard-boiled egg and you drop it into a bowl of water that has color added to it. Well, where does the color come from? Typically, it comes from, you know, you buy a Easter egg coloring kit at the supermarket. It's got a little colored dye tablet in it. It's pink or purple or yellow or green. You drop the tablet into clear water. The tablet dissolves and the pigments of the tablet spread out through the volume of water to color the water. The spreading out of the pigments from the tablet. Hey folks, that is simple diffusion. That's all it is. Spreading out of the dye pigments, okay? So how would we define simple diffusion? Okay, simple diffusion can be defined this way. This is the movement of a substance. Well, what do I mean by substance? That substance could be individual atoms, it could be ions, it could be molecules, it could be particles. And by particles, I mean potentially groups of atoms or even groups of molecules that are lumped together as an entity. So, okay, so it could be particles. It could be something we consider as a solute, right? Remember we talked about a solute, right? A solution equals a solvent plus a solute. The solute is the dissolved material. So solutes can move. And honestly, the solvent, you know, the solvent in biological systems is going to be water. The solvent can move from an area of high to low concentration, okay? So simple diffusion is the movement of a substance, whether that substance is an atom, an ion, a molecule, particle, something we would describe as a solute, something we would describe as a solvent, from an area of high concentration to an area of low concentration. Okay. And again, the video tried to show you some of these things. Please stop me along the way if you have questions. Please ask questions. Unmute your microphone. Type into the chat window. That's quite okay. Okay. So, you know, there's lots of things we could say here. So an example of cargo that is moved through the plasma membrane by simple diffusion, I'll just give you two of them right now, okay? Oxygen, O2, and carbon dioxide, CO2. These are tiny molecules that are basically uncharged. And so they can float through the narrow spaces in between phospholipids and get right into the cell or out of the cell. Okay. And think about it for a moment. Think about the volume of air that you just inhaled 10 seconds ago, right? You brought it into your lungs. There was a tremendous number of oxygen molecules in that air that you just took into your lungs when you inhaled. Somehow that oxygen has got across the cell membranes of the anatomy in your lungs that merge with your blood vessels to get the oxygen into your blood supply. So the oxygen can be delivered to your brain and down to your big toe throughout your entire internal anatomy. Isn't it a really good thing? Isn't it a really good thing that our cells do not really need to burn any ATP to move that oxygen? Because if we had to burn ATP to move oxygen, I suspect we would barely have enough energy to actually be alive. In other words, we would use all of our available energy simply to move oxygen. We wouldn't have any energy left over to take a college course or to drive a car or to play baseball or watch a television show, okay? So it's very nice that many of these pieces of cargo are passively moved through the membrane, because if we had to burn our own energy all the time for it, things wouldn't go too well. Okay, any questions? Okay, well, simple diffusion is the first type of passive transport process. Next, we have facilitated diffusion. Okay, facilitated diffusion is talked about in its various forms, honestly, on figures 3.5 and 3.6 and 3.7. Okay, so you can look at all of them. Okay, the definition, the general definition for facilitated diffusion is the movement of a substance, okay, across a membrane with the assistance of a protein. The movement of a substance across a membrane with the assistance of a protein from high to low concentration, right? Notice the common language in these definitions. Simple diffusion, high concentration to low concentration. Facilitated diffusion, from high to low concentration, same thing. Okay. Now with facilitated diffusion, there's two major types because there's two different kinds of cargo that need two different types of protein to move, okay? So we have subtypes of facilitated diffusion. We have channel-mediated facilitated diffusion. And in channel-mediated facilitated diffusion, right, that will be the topic of figure 3.6. What you will notice is that the protein is moving some kind of ion. Channel-mediated facilitated diffusion is used to move ions, like sodium, potassium, right? Chloride, etc., etc., etc. Again, from high to low. In addition to channel-mediated facilitated diffusion, we have carrier-mediated facilitated diffusion. This is going to be used not to move ions, but to move larger molecules that still have a little bit of polarity to them, okay? So we have carrier-mediated. Figure 3.7, okay? Used to move molecules, you know, certainly something like glucose would be larger than an individual ion like sodium, but as molecules go, glucose is still a pretty tiny molecule, okay? We'll have to talk more about that in the next 10-15 minutes. So carrier-mediated used to move molecules like glucose through the plasma membrane from an area of high to low concentration. Again, the cell is not consuming ATP to make this process happen. All right. Do you have any questions right now? Let's keep rolling here. All right, third form of passive transport is osmosis. Osmosis is an extremely important concept for future healthcare workers, particularly nurses, to appreciate, okay? Because if you have a patient that is overhydrated or dehydrated and you apply the wrong IV, you could actually harm your patient, and it's because of osmosis, okay? So this is a deeply important topic. Your book talks about it with language on page 71, and there's some visualization of principles on page 72 and 73. Okay. So let's see here, figure 3.8 and figure 3.9 are relevant to you here. Okay, there's different ways we could talk about this. Okay, and so I'm just going to say a couple things here. Okay, osmosis is referring to the motion of water across the membrane. Okay. And there's really some nuance here, okay? As your book points out on page 71, the column of text, osmosis can move water through the plasma membrane by two alternative mechanisms, okay? In some cases, water is moved by osmosis. In some cases, water is moved by osmosis that utilizes simple diffusion. Okay, so in some context, osmosis can be considered a subtype of simple diffusion. Okay, in other cases, water is moved by osmosis through membrane proteins called aquaporins. Okay. And so if there's a membrane protein that's involved in moving the cargo, that would be a subtype of facilitated diffusion, right? The membrane protein is facilitating the motion of the water. So you might say, well, why don't we just talk about the movement of water as simple diffusion or facilitated diffusion? And you can make an argument that that would be an appropriate conversation. But for historical reasons that are not always obvious, we tend to bring out the topic of water motion as osmosis and talk about it separately. Whether that's a good thing or bad thing is a philosophical question. Okay, so some other things here. Okay, the important thing to appreciate is that, you know, whether you think of osmosis as a special type of simple diffusion or a special type of facilitated diffusion, we need to understand how water is moving, why it is moving. That's the bigger issue here right now. So I'm going to give you what I like as a definition of osmosis, okay? Osmosis is the movement of water across a membrane from an area of high free water concentration to an area of low free water concentration. Okay, so unless you've really had some science previously, I suspect this is pretty new language to you, and critical to understanding this is this concept of free water. What in the world do I mean by free water? Okay, so first question here, question A. What is meant by free water? Okay, can you answer that in the chat window, folks? What do I mean by free water? What do I mean by free water? What circumstance would make water not free? Okay. All right. Thanks for everybody that's starting to put some comments down. Is it free moving? Well, if it's free water, it is free moving in a sense, Rachel. That's not bad. No, water that isn't in something. Well, water has to be in something, doesn't it? If anything, it has to be in some physical space. Shay, you say no chemical bond. I really like that. So let me run with that. What I mean by free water is a water molecule that is not interacting with a solute. A water molecule, which is solvent, that is not interacting with the solute. Okay. This refers to water molecules, which again would be solvent, that are not interacting with solute. Okay. So Rochelle says, so it's just water, nothing added. That's true. It would be very pure water. Okay, now most sources of water on our planet are not naturally completely pure, right? If you take water even from spring water, there's all going to be all kinds of dissolved minerals and things in there, which gives water some taste when you drink it. Okay, so when I say free water, I'm talking about water that is not interacting with solute. So with that new understanding, let's reread the definition. Osmosis is the movement of water across a membrane from an area of high free water, where there's a whole bunch of pure water and very little to no solute, to an area of low free water, where there's a bunch of water, but it's all interacting with solute. That's how water is going to move. Okay, that's the direction that water is going to move. So my next question is this. What property of water explains why it, meaning water, often interacts with other substances, meaning solutes? Okay, what property of water explains why it often interacts with other substances? What property explains this? Okay, thanks, Rochelle. That's not quite there, but I appreciate you trying. Okay, Rachel hit upon it. Rachel hit upon it. It's polar. Water is a polar substance, right? It has an end where you have a partially positive hydrogen atom, and the opposite end where you have a partially negatively charged oxygen atom. Water is a polar molecule. Those partial charges create circumstances that allow you to interact with other solutes that have some kind of electric charge. So if you have a solute with any electric charge, positive or negative, it will be attracted to one or the other end of water. And if that's the case, then that water is no longer free. Okay. So this is a topic I could spend the rest of the morning on, and I don't want to, but here's what I would like you to do during your private study time. During your private study time, I want you to go to YouTube, and when you're in YouTube, I want you to type in this phrase, how osmosis works. How osmosis works. And let me show you what happens when I do that on my computer. Come on, computer, do something. So when I go to YouTube and type in how osmosis works, right there it is. You're going to get this very first video, 1 minute, 43 seconds. It's a great video, and it explains how simple diffusion and then osmosis actually happens. Okay, so see if that is helpful to you during your personal study time. Okay. All right, folks, any thoughts or questions you need to ask right now? Okay, let's go. Let's keep rolling here. It's never enough time. Okay, so next, I'm going to talk about active transport processes. Okay, active transport processes. Active processes require the cell to burn energy, which is usually ATP, to move cargo. Number two, active transport processes are typically used typically used to move cargo against their concentration gradient. In other words, rather than moving cargo from high to low concentration, we will move it from low to high concentration. Usually used to move cargo from low to high concentration. Okay, now there's different forms of this, and we're not going to be able to talk insanely about all this stuff, but I can highlight them for you. First, we have active transport. Okay, active transport is the conversation in your book begins at the bottom right corner, page 72, and it goes over to page 74, 75, 76, 77. These are the pages that I need to talk to you about. Okay, active transport uses cellular energy to move cargo from low to high concentration. Okay, and there's basically two subtypes here. Here's what they are. Okay, we have primary active transport. Primary active transport. This is what figure 310 is trying to share with you. Okay, so a beautiful example of primary active transport is the sodium-potassium pump. Okay, and in this scenario, energy is released from the breakdown of ATP. If you're looking at figure 310, look at the second panel where you see blue ball number 2. Blue ball number 2. Don't you notice a molecule of ATP is being broken down into ADP? So in primary active transport, energy released from the breakdown of ATP is used to quote unquote pump cargo through the plasma membrane. Okay. The second type of active transport is the secondary active transport. Oh boy, how much time we got here? 9:09. My God. All right. Tell you what, folks, here's what I'm going to do. Since we do have lab today, I'm going to slow down my roll a little bit. I'm going to take time to show you a video on this now, and then we will finish the note taking in lab. That way you have some visualization to think about this while I'm giving you notes, okay? So let me show you the second video on active transport processes. We'll finish our notes in lab today. That should not be a problem. Okay, here we go. Okay, we'll cue up the second video, which is membrane transport active processes. Just enjoy. You can watch this again at home. Here we go.
The 60 to 100 trillion cells of the human body are surrounded by extracellular fluid containing substances needed by the cell, such as oxygen, ions, nutrient sources of energy, and ingredients for cellular functions. This fluid is also the location for disposal of wastes and carbon dioxide. All of these substances need to be transported across the cell membrane. Recall that passive processes include diffusion, facilitative diffusion, and osmosis. These processes occur as a result of concentration gradients of the substance, thereby requiring no energy source to move the substance from one side of the cell membrane to another. Active processes utilize energy to move substances across the membrane, generally from areas of lower concentration to areas of higher concentration. Active transport involves protein transporters known as pumps. Other active processes use membranous transport vesicles. There are two types of active transport, primary and secondary. Primary active transport requires energy derived from ATP. An important primary active transport mechanism involves the movement of sodium and potassium across the plasma membrane using a sodium-potassium pump. 3 sodium ions in the cytosol attach to binding sites in the pump. This triggers ATP to bind to the pump and split into ADP and a single phosphate. The released phosphate attaches to the pump, causing it to change shape and open to the opposite side of the membrane. The affinity of the sodium binding sites decreases, causing the release of the sodium ions into the extracellular fluid. The changed shape of the pump now increases the affinity of the potassium binding sites, and 2 potassium ions are drawn into the pump and attached. This action releases the phosphate, and the pump returns to its original shape. The potassium ions are expelled into the cytosol as sodium ions enter the pump, and the process continues. Secondary active transport uses the energy released when one substance moves down its concentration gradient to move another substance, such as glucose, against its electrochemical gradient. The driving force is primarily the movement of sodium ions from areas of high concentration to areas of lower concentration. When 2 substances are transported in the same direction by secondary active transport, the protein transporter is known as a symporter. Glucose molecules are co-transported with sodium ions from the extracellular fluid into the cell. The occupation of the glucose binding site causes the symporter to change shape, exposing both substances to the cytosol. When the sodium ions move down their electrochemical gradient into the cytosol, glucose is released into the cytosol against its concentration gradient. Antiporters are protein transporters that move solutes in opposite directions. For example, hydrogen ions are transported out of the cell against their concentration gradient as sodium ions move through the same antiporters down their electrochemical gradient into the cell. This movement of hydrogen ions is important in the regulation of blood pH in the kidneys. When larger particles or greater quantities of solutes need to be transported across the plasma membrane, another active process is utilized. Vesicular transport involves the formation of solute-containing spheres known as vesicles. The types of vesicular transport are identified by the direction of the movement. Vesicular transport from the extracellular fluid into the cell is termed endocytosis. Exocytosis refers to vesicular transport out of the cell. All cells secrete materials such as hormones, digestive enzymes, or neurotransmitters via exocytosis. A membrane-enclosed secretory vesicle in the cytosol fuses with the cell's plasma membrane, and its contents are released into the extracellular fluid. Endocytosis occurs in 3 ways: phagocytosis, receptor-mediated endocytosis, and pinocytosis. Phagocytosis occurs when cells, known as phagocytes, engulf other cells, bacteria, viruses, and even indigestible particles. When the unwanted material binds to receptors on the phagocyte's membrane, it and the cytosol extend to form pseudopods that surround the unwanted cell or particle and eventually engulf it completely in a vesicle called a phagosome, transporting it into the cytosol. There, the phagosome will fuse with a lysosome, with enzymes that attempt to digest the consumed material. Receptor-mediated endocytosis occurs in cells containing specific protein receptors on the surface of their plasma membranes. When molecules such as hormones or antibodies that the cell needs attach to the receptors, it causes the membrane to cave in and surround the molecules. The receptors are returned to the cell's surface and the molecule is released into the cytoplasm. In a third type of endocytosis, known as pinocytosis, the plasma membrane caves in to form a pouch that fills with droplets of extracellular fluid. After it is sealed, the vesicle separates from the membrane and passes into the cell, where lysosomes break down the contained solutes for cellular use. When a vesicle carries substances from one side of the cell to the other, employing both endocytosis and exocytosis, the process is called transcytosis.
Okay, folks, so there we've done it, so to speak. We are out of time, so I'm going to stop sharing. We will finish this topic in lab. I think I need 25 minutes to do it, but we have the time to spare. We'll do that. We'll get into lab, answer questions, get you ready for your lab exam next week. Okay, so let's pause at this time. Let's start lab at 9:35. The Zoom window will stay open. You can stay logged in, okay? See ya. Take a break. See you at 9:35.
Thank you.
Okay, welcome back, everyone. Hope you had a little moment to refresh, maybe get a little snack or something. Now let's see if we can come back together and finish our conversation from our 8 a.m. lecture. Okay. So I was rushing a little bit. I decided to slow it down. We'll just finish our note taking here in lab. We have plenty of time. We can do this. Be sure you get good coverage of the content. Okay. Do you have any questions that you would like to ask from the video we saw right before we went on our break? Okay. So what I'm going to do at this time is finish the note taking on those processes. Make sure you have a basic definition of them in your notes. And then we're going to do a practical example where we try to apply our knowledge of the variety of transport processes that we've learned this morning, okay? All right, so I'm going to share my screen with you again. All right, and we're back up, ready to go. Okay, so once again, active processes, that's what we're talking about right now, active processes. Active processes require the energy, require the cell to burn some type of energy. It's typically ATP to move the cargo. It often moves the cargo from low to high concentration, although there's some cases when you actually move high to low, but you still need energy because the cargo is so very large. That's more of an endocytosis business. Okay. So the various types of active transport processes include active transport. There's two subtypes: primary active transport, figure 310. You release energy from the breakdown of ATP to move cargo across the membrane. And you see the four panels of the sodium potassium pump. You're going to encounter the sodium potassium pump again in our semester when we talk about neurons. Okay, so it's going to come up again. Secondary active transport is highlighted in figure 311. Okay, and in secondary active transport, you have the energy stored in an ion gradient. The energy of moving the ions through the membrane powers the motion of a second substance in the opposite direction. Okay, you see how, as in figure 311 on page 75, that as sodium comes in, calcium is pushed out, or as sodium is coming in, hydrogen is pushed out. Okay, so we have secondary active transport that will move cargo in opposite directions. We do have secondary active transport that will move cargo in the same direction. That will be the right side of figure 311, where sodium and glucose, their motion is coupled in the same direction. Okay, so let me get a basic definition into your notes. The energy stored in a concentration gradient, like sodium or hydrogen, is coupled to the motion of a second material. Coupled to the motion of a second material. Okay, this is going to use membrane proteins, right? You can see they're color-coded. The membrane proteins that move cargo in the opposite direction are color-coded blue on figure 311. The membrane proteins that move cargo in the same direction are color-coded yellow. But the idea here is that the motion of one substance is coupled to the motion of a second material. Okay, so that is the two forms of active transport. Okay, the cell will burn energy to make that happen. There are other types of active processes, okay, and the main talking point for the next conversation is something known as vesicular transport. A vesicular transport, as a heading, is on the bottom right panel, page 75, and it spills over to the next several pages. Okay? So when we're talking about vesicular transport, there are several types. Okay, let me do this. We have endocytosis. Okay, and within endocytosis, there are three primary types. Okay, hold on a second. I need to respond to a student in the chat window here. Okay. Okay, and we'll continue. So endocytosis. Endocytosis comes in three major flavors, if you will, three major types, right? There is receptor-mediated endocytosis. That would be what you're seeing with figure 312. We also have phagocytosis, right? In the video, you saw phagocytosis. Phagocytosis is a process carried out by cells that are named phagocytes. They are white blood cells. They are defense cells of the immune system, and their job frequently is to defend your body against bacteria, viruses, other invaders of the body that might seek to cause us harm. So that's the topic of 313. Okay. And then we have pinocytosis, which is also known by the phrase bulk phase endocytosis. That's figure 314. Pinocytosis, figure 314. All right, so there's lots of good stuff for you to study, also known as bulk phase endocytosis. Now, you certainly might ask yourself, why are all these different processes necessary? Why did Mother Nature create all these different processes? And it's a very reasonable question that I don't think we would ever have an answer for. In other words, sometimes the logic of how various processes have evolved over large spans of time really isn't available. We can't figure out why Mother Nature did it this way. In other words, if we were going to, as human beings, engineer the human body, we would probably engineer it differently than Mother Nature did. So the logic of how or why Mother Nature created pinocytosis versus secondary active transport versus anything else often is something that we can only philosophically guess about. Okay. All right. So the big deal about endocytosis, whether it's receptor-mediated phagocytosis or pinocytosis, is that you're moving cargo into the cell. And Endocytosis is typically used to move large cargo. For example, okay, if it's phagocytosis on page 77, figure 313, right? You have a white blood cell that's ingesting an entire bacterial cell. That's very, very large cargo. So the type of cargo that's typically brought in by all three forms of endocytosis is actually quite large and made of honestly billions of molecules moving as a group and being internalized as a group. Okay, so endocytosis is used to bring large materials in large quantities of molecules into the cell. All right, I'm going to let you wrestle with the details of these things because, well, we have so much to do. Okay, so endocytosis, you bring large quantities of materials into the cell, all right? Exocytosis is the other type of vesicular transport. Okay, exocytosis is the opposite of endocytosis in the sense that you are pushing large quantities of material out of the cell. Okay, so endocytosis, we move in. Exocytosis, we move out. Okay. And then there was a third form of motion that was listed in the video today. And it is talked about in your textbook, but I don't think there's a picture of it. And that is transcytosis. Transcytosis is the movement of large cargo or large quantities of material into the cell through the interior of the cell and out a different surface of the cell. So simplistically, it would be into the cell, across the interior, and out the other side. If you couple endocytosis and exocytosis together at the same time, we call that transcytosis. This is the movement. This is the coupling of endocytosis with exocytosis. Okay. In other words, material is brought into the cell, moves across the interior of the cell, and exits the cell. Okay, now that might seem like an odd thing, but I'll give you an example of that in just a moment. Okay, all right, gang, we have done our due diligence here this morning. We have at least got these processes defined and referred to in your notes. Obviously, you're going to have to spend some more study time really thinking about this content. There is one more thing I want to do with you this morning to help get these ideas into your head. But before I do that, do you have any questions? Okay, everybody's so quiet. I don't know if that means you're passed out or what's going on, but I hope you're okay. So let's do this. As a way of trying to clarify these topics, I want to make a drawing with you. And I'm going to ask you to please draw with me at home. If you have availability for a couple different colored pens or pencils and a sheet of blank paper, that would be very nice. And I'm going to insert a new page into my document here. And I'm going to start drawing. Okay, but before I do that, let me give it a title heading. So we're going to do a practical application of cargo transport through the plasma membrane. So we're going to try to make a fairly real world scenario here and see if we can understand it. Okay. All right, so here we go. I'm going to start drawing, and I apologize right off the bat because I am not much of an artist, but hopefully you can get the general idea of what I'm trying to convey to you. So, okay, here's what I'm going to propose. Okay, we're going to talk about blood sugar regulation in the context of insulin release, okay, because this will involve a lot of transport processes. So, first off, I'm going to draw what is supposed to be a blood vessel. Okay, so here we have a blood vessel. I want to encourage you to draw along with me. Okay. And so we're going to place this blood vessel actually inside an organ. And that organ for diabetes regulation, for sugar regulation, is going to be the pancreas. So I'm just, again, I'm not an artist, but I'm just going to draw sort of a big blob here and say, okay, this is part of the pancreas. And of course, the pancreas as an organ is made of many cells. So here's a cell, right? All right. Now, the blood vessel that's going to be in any organ, if it's truly in any organ, it's most likely part of a capillary bed. So this blood vessel is going to be a capillary. Okay. And so this up here is the capillary wall on, both the top and bottom are part of the capillary wall. If you asked me to draw this in 3D so it was more realistic, I would pass out because I can't do that. But the idea here is with this blood vessel, you should be visualizing a cylinder, a tube, right? It's a blood vessel. It's like your garden hose. It's like the interior cardboard cylinder of a paper roll, of a paper roll stack that you have, paper towels, right? Inside of paper towels. So remember that this capillary wall is a cylinder, and we're kind of looking at the top of the cylinder and the bottom of the cylinder here. And notice that the cylinder wall is actually made of a network of cells. Okay, so the capillary wall is made of cells. We have a blood vessel here that's part of the vasculature of the pancreas organ, and we have a pancreas cell defined. Okay, so obviously, inside the blood vessel, we should find blood. Okay, so we have blood that's moving along here inside the blood vessel. And what I want to say is, okay, so let's start with something practical. Okay, you probably had breakfast this morning. I hope you had breakfast this morning. So you probably had a bowl of cereal, maybe you were up early and you had some pancakes, who knows what, but you ate some food, which means the sugar level, the glucose level in your bloodstream is going to go up because you ate a meal. So we're going to put into the bloodstream some glucose, okay? Glucose. Now, we know that the liquid inside the blood vessel is the blood, but there's also going to be liquid outside the blood vessel. In other words, there's not empty space inside the body. So I'm going to ask you at this time, what is this space? What is the liquid in this space called that I'm putting orange brackets around in the drawing at this time? What is that? What is it called? Do you know? Okay. All right. Shay, thanks for answering, and I appreciate that because it lets me help you. The plasma would be in the blood vessel, right? Blood plasma is inside blood vessels. It's not outside blood vessels. So it's not plasma, not really, but it's something related to plasma. Does anybody know? So when you have liquid that's outside the blood vessels, it's outside the blood vessels, it's outside the organ. It kind of fills up the space between the blood vessel and the cells of the organ. This is known as interstitial fluid. Okay, Noah, you said something equivalent. Right, this is interstitial fluid. Okay, Shay, you said plasma. That's not technically correct. The plasma is in the blood vessel, but let me mention that for everybody's benefit, that the interstitial fluid is created by filtering the blood plasma. And we'll learn, you'll learn about that in A&P too. Okay, so we have the blood in the blood vessel. We have the interstitial fluid, which is really filtered blood plasma outside the blood vessel. All right. So here's what I want to do is I want to trace the motion of glucose from the blood vessel to the pancreatic cell and get you to tell me what the transport processes involved would actually be. Okay, so here we're going to start here. I guess I'll stay with my orange color. So what I want to do is move this glucose molecule from here, location A, to location B. So folks, in the chat window, I'm going to ask you A to B. I want you to tell me the name of the transport process that will be used to move glucose from A to B. What's the name of the transport process to move glucose from A to B? What's it called? What's this going to be? We learned a lot of different processes today. Okay. All right. Okay, so Noah, it is not exocytosis. Okay, what else could it be? So Noah, for this to be exocytosis, you appreciate that the glucose would have to be inside a cell and exiting the cell. Right now, the glucose is not inside a cell, it's in the plasma. It's not inside a cell, it's in the plasma. Okay. So, but suffice it to say that we got to take a glucose molecule and get it through the plasma membrane of these cells that are making up the wall. Okay. So what should this be? Any takers? Did we talk about glucose in any specific scenario today in our notes? Okay, Marie, you said carrier-mediated diffusion. You are right, and Marie, be very complete. You are right, but on an exam, make sure you say the whole thing. It's carrier-mediated facilitated diffusion, right? All right, so A to B would be carrier mediated facilitated diffusion. Okay, so let's think about Noah's answer of exocytosis and why that was not correct. Okay, this is not exocytosis because first off, exocytosis is used to move very large cargo or groups of molecules. Well, in this case, glucose is not being moved through a bubble scenario. It's simply free-floating individual glucose molecules. So they have to move through carrier proteins embedded in the plasma membrane of the cells making up the capillary wall. That's always going to be a facilitated scenario. And as Marie correctly put in, it's going to be carrier-mediated facilitated diffusion. Okay. Question in the chat window. Is this making sense so far? Okay, what questions do you have? Okay, all right. Let's continue here. So let's say the glucose is now at location B, and I want the glucose to go to location C. So I'm going from I'm running out of space, but I'm going to go from B to C. So in the chat window, we're going B to C. What is the transport process? If the glucose is moving from location B to location C, what is the transport process? What do you think, folks? Come on. I'm not going to give you this one. You got to think it. Okay, Rachel, thanks for participating. This is not transcytosis. It is not transcytosis because the location that the glucose is in has no cells. There's no cell here. It's simply the interstitial fluid. Okay, Shay, you say carrier-mediated facilitated diffusion. Shay, this is not carrier-mediated facilitated diffusion because unlike moving from A to B, when we move from B to C, there is no membrane. There is no membrane. From B to C, there is no membrane. So this cannot be a facilitated process. Okay, so what else could this be? There's one process that we talked about today that does not require the presence of a membrane to take place. What was that process? Give me some options, folks. Come on. Okay, thank you, Brittany. You're not correct, Brittany, but I appreciate you participating. Okay. Anybody else? Let me give you a hint. Take a look at figure 3.4. Figure 3.4. What process is being illustrated in figure 3.4? Okay, Rachel, it's not facilitated. There's no membrane here. Okay, so Shay, you say diffusion. Now, in terms of the language of our conversation, we have facilitated diffusion and simple diffusion. So Shay, which version of diffusion is it? Okay, so there we go, folks. This is simple diffusion. Why the heck is this simple diffusion? Again, do you see any membrane between the location B and location C? There's no membrane. There is no membrane, so there's nothing to facilitate. It's simply like having a beach ball floating from one side of your swimming pool to the other side of your swimming pool. There's just the water. There's just the interstitial fluid. Okay, so B to C is simple diffusion. Question in the chat window. Is it becoming clearer why A to B and B to C are different? Is it becoming clearer why they are different? Any questions? Okay. Okay, so let's continue here. Let's say I want to take the glucose from location C And I want to get it across the plasma membrane of this pancreatic cell and go to D. Okay. So let's do C to D. In the chat window, what would be the transport process of C to D? Okay. I love you folks, and I appreciate your participation. No, it's not endocytosis. It's not endocytosis. Why is it not endocytosis? Because endocytosis is used for very, very large substances or volumes of many molecules at a time. We're moving a single molecule right now. This is not endocytosis. Endocytosis would not be used for a single molecule. Okay, so this is facilitated diffusion. Again, it's carrier-mediated facilitated diffusion. It's the same thing as A to B. Okay, so C to D is carrier-mediated. Facilitated diffusion. Okay, so Noah, you made a comment on the chat window. I wasn't aware of the size. That's a very important realization. Thank you for saying that, because that's key to understanding this question. Okay, so folks, I never have enough space here, but what I would like you to do is give me the molecular formula. Please give me the molecular formula for glucose. Can you get it and type it in the chat window? What is the molecular formula for glucose? Okay. If you don't know what molecular formula is, fine. Well, just Google it. Type in molecular formula glucose. See what comes up. Okay, I'm going to go with the first answers, right? C6H12O6. That's the molecular formula. Okay. And so for Noah's benefit and everybody's benefit, what this is telling you is that a molecule that has what? How many atoms? 6 plus 12 is 18. 18 plus 6 is 24. So we have 24 atoms here. So this molecule is made of 24 atoms, and so it is the right size for carrier-mediated facilitated diffusion. This is the size of molecules where you will use a transporter protein to move that cargo across, something on the order of multiple tens of atoms, like glucose, 24 atoms. That's the right size for a facilitated diffusion process. Okay. Now let's continue here. Remember, we're talking about this in the context of the main point here is about transport processes, but I'm using the concept of blood sugar regulation and metabolism to illustrate these principles. So when that glucose finally enters the pancreatic cell is going to be aware of a couple things. The fact that glucose has entered the cell is going to tell the cell that blood glucose has gone up. Blood glucose has gone up. How does the cell know? Because it just received glucose from the bloodstream. So it infers, yeah, blood sugar level just went up. And so how will the pancreas respond to an elevation in blood sugar? Well, it's going to release insulin. So it's possible that there is some insulin that's already been made by the pancreatic cell and it will be released. And it's also very likely that the pancreatic cell will begin making new insulin. So blood glucose has gone up. That's one thing that will happen in the interior of the cell, will be the realization of that. And number two will be, let's make more insulin. So the pancreatic cell is going to make some insulin. The process of how it makes insulin is going to be a major talking point for next lecture. I can't cover it all today, it's too much. So suffice it to say, insulin is going to get made in the pancreatic cell, and I'm going to highlight insulin by the letter I with a triangle. Okay, this is insulin at this time. And so what we need to do is get insulin from inside the pancreatic cell, which I'll make this, that was ugly, wasn't it? I'll make this location E. So insulin is now at location E, and I need to get insulin to location F. So, folks, let's do this. Chat window, E to F, E to F. What's the transport process that's going to be involved in moving insulin out of the pancreatic cell into the interstitial fluid? You got to take insulin inside the pancreatic cell, push it out of the cell, get it into the interstitial fluid. What transport process is going to be involved? Some of you are probably gun shy because I keep saying, no, that's not right, but just try. Okay, I'm going to go with the first response on the chat window. Karen, this is exocytosis. Yes. Let's write it down, then we'll explain why it's exocytosis. Okay, so let me do this. E to F, this is exocytosis. Okay, so why is it exocytosis? This is exocytosis because the molecular formula of insulin is just what Karen typed into the chat window. Because beautifully done, Karen. That's exactly what I was going to ask next. Okay, so let me write this down. The molecular formula of insulin is in fact C257, hydrogen 383, nitrogen 65, oxygen 77, sulfur 6. Okay, and so let me see here. We got to add those up. I'm going to use my calculator for that. So we've got 257 plus 383 plus 65 plus 77 plus 6. That's 788 atoms. Okay. 788 atoms. Okay, think about that. What I'm trying to show everybody is that insulin, just based on the total number of atoms that are in that molecule, is tremendously larger than glucose. And because it is tremendously larger than glucose, it is too large. It would be too large to fit a protein through another protein embedded in the membrane, right? It's like trying to fit a hammer through a hammer. That just doesn't make any sense. Okay? So because the nature of the cargo is much larger here, and we've got to get through the membrane, and we can't take a protein through another protein, that's silly. We have to use an alternative way of getting the cargo through the membrane, and the most logical way to move large cargo or large volumes of cargo at the same time is going to be exocytosis. Okay. Question in the chat window. Is this making sense? Are there any questions you'd like to ask? Okay. All right, so Israel, you said it's making sense a bit. So what can I help you with? Is there anything you'd like to ask Israel for the benefit of everybody? Okay, so you say you need some more study time. That's a fair statement. Okay. All right. So, when you think about the motion of material through the plasma membrane, or the motion of material anywhere, first off, if you're thinking about the motion of material, you have to ask yourself, is a membrane present in between the two locations? If there's not, that's immediately simple diffusion. If there is a membrane, then you need to think about the size of the cargo. And by size, I mean how many atoms make up the molecule. And you have to think about whether the molecule has an electric charge or not, right? Because even a small ion, a single atom with a charge like a hydrogen ion, is going to need a facilitated process to move across the membrane. Okay, let's continue here. We'll wrap this up fairly quickly. So we want our insulin at location F to go now to location G. Okay, G is still in the interstitial fluid. So now I want to ask everybody in the chat window, what is the method of transport from F to G? F to G, what is it? All right, Kelly, seems that your things are clicking for you. And Kelly is absolutely right. You see your answer in the chat window. F to G is simple diffusion. Why is it simple diffusion? It was just exocytosis a moment ago. Well, again, we're moving the cargo currently between one location to another location where there's no membrane. There's no membrane. That's simple diffusion. Okay, so F to G is simple diffusion. Okay.
So, basically, that simple diffusion isn't a membrane, and the exocytosis is when there is like a bigger mass.
Rochelle, exocytosis is definitely when there's a bigger mass. That's true. Simple diffusion as a process, depending on the nature of the cargo, may or may not use a membrane. But if you see any cargo moving from one location of a liquid to another location of a liquid and there's no membrane in between, that will always be simple diffusion.
Okay.
Okay. Any other questions? All right, so let's take the insulin now from G into H, where we get it into the bloodstream. Now, this is going to be more complicated, but let me just throw it out there. If you can explain to me or if you can at least give me the answer. So from G to H, you have any idea what the transport process would be from G to H? Any guesses? Just take a guess. It's okay to be wrong, right? It's okay to be wrong right now because this is not an exam. Nobody wants to take a guess? Okay, thanks, Israel, for taking a guess. I'm getting lots of answers here, aren't I? Carrier-mediated facilitative diffusion, simple. How about endocytosis? Lots of good stuff here. Unfortunately, technically, there's no correct answer on the chat screen yet. Folks, this is going to be transcytosis. You might say to yourself, what? Transcytosis, how did he come up with that? I'll explain it to you in just a moment. G to H is transcytosis. Okay, now to understand this, we got to do some additional stuff here. Israel, I see your comment, and I'm going to defer answering it for the moment. Okay, but it's a very good question. Okay, so hold on tight. Okay. What I want to highlight here is this. Okay, to answer this question of G to H, we need to focus on the anatomy of the blood vessel wall at the location that I'm highlighting in blue. So what I need to do is redraw this anatomy in a magnified, enlarged way so that this makes sense. Okay, so I'm just going to give you 5 seconds to look at this drawing. And then I need to move it off the screen because I got to draw something else. So we're going to take the area that's highlighted in the blue rectangle and we're going to redraw it in a larger way down below. Okay, so here we are. So I'll just put down C below. Okay, all right. So I'm just going to make just a silly little dividing wall here so you know that we're drawing something else. Okay, so I need to redraw that section of wall and make it larger. And so here's what I'm going to do. Okay, so we have the top of the blood vessel wall here. But remember, this is really a cylinder. It's like having a cylinder from Christmas wrapping paper in your hand, and you're pointing to the top of the cardboard cylinder versus the bottom of the cardboard cylinder. So this is the top of the capillary wall. And now the bottom of the capillary wall, I'm going to draw it differently to emphasize the anatomy that I need you to see at a microscopic level. Remember, the capillary wall is made of cells. So what I really mean is that the capillary wall is made of cells, and I'm going to draw the cells as these large rectangles. These are cells. Okay, so these are cells that make up the capillary wall. All right, and what I want to highlight to you is, right, we were talking about having insulin in the interstitial fluid get into the bloodstream. So what that would really involve is the following. Insulin would, let me use a different color here. Insulin would enter the cell by endocytosis. It would go across the interior by being dragged along by cellular machinery in the cell, and then it would exit, right? So we enter, we go across the interior, and we exit. We have coupled endocytosis and exocytosis, which is why insulin going through the cells of the capillary wall and actually gaining access to the blood supply is transcytosis. So is endocytosis involved? Yes. But from the perspective of viewing the entire motion of the insulin through the capillary wall into the bloodstream, we've coupled endocytosis with internal motion of the molecule through the cytoplasm of the capillary cell and out the other side of the capillary cell, which is exocytosis. You put it all together and it is transcytosis. Okay. So question in the chat window. Is this okay with the new drawing? Is it making sense, or you still don't understand? Is there anything you'd like to ask? Okay, Israel, I haven't forgotten about your question.
So even though it's not like going to, like it's going out of a cell to a not cell, that's still transcytosis?
In this case, yes, because we are coupling all three processes together. No, we are first starting with endocytosis to get the insulin into the cell. Then we're moving that insulin cargo through the interior volume of the cell. Then we're moving that cargo out the other side of the cell. When we couple endocytosis with the exocytosis, then we refer to it as transcytosis. Why would this not be exocytosis all by itself? Because insulin was not made inside the cell it's passing through. It was not made inside the cell it was passing through. So it's not just exocytosis. Other questions? Okay, I want to come back to Israel's comment in the chat window. We were talking about the motion of insulin, which I've just described as transcytosis. And she asked, so AB, so wouldn't GBH be the same? She's asking, wouldn't the motion of A and B and G and H be the same? No, Israel, it wouldn't, because in A to B, we're moving glucose, which is a tiny molecule. So that's facilitated diffusion, moving through a protein. With insulin, it's much larger. I can't take insulin protein and shove it through another protein. So that's got to be a vesicular transport process. That's why that's going to ultimately be transcytosis. Okay, so A to B is moving a small molecule. G to H is using a gigantic whale-like molecule. Okay. All right. Other questions you'd like to ask? Do you think you could do this for me on an exam? Think about it. Study it. Think about it. Study it. Watch videos. Okay? You can do it. All right, gang. Any questions before we stop what we're doing here with lecture? Okay. All right, then I'm going to stop sharing my screen momentarily. I'm also going to stop recording because we're done with lecture.